Hello and welcome to today's webinar, Extending a Trial's Design, Case Studies of Dealing with Study Design Issues Demonstrated on nQuery. So today's webinar will be looking at some common issues that occur in clinical trials and field trials where standard statistical methods or designs aren't appropriate and how you could extend upon those standard methods to get a more appropriate statistical analysis for your design and the implications that would have on your sample size determination. So before we get started, just a few frequently asked questions. Firstly, is this webinar being recorded? Yes, it is being recorded, and the slides and the recording will be sent to you after this webinar is complete later today. Secondly, if you have any questions that you want to ask about anything in this webinar, please feel free to use the questions tab on the right-hand side of your webinar software and enter them there. I'll try to answer as many of those as possible at the end of the webinar. And any that I don't get around to, I'll make sure to email you with an answer either today or tomorrow. Okay, I think that basically covers that. But if you have any issues with the audio or any questions, do let me know using that chat window. So as said, today's webinar is Extending a Trial's Design, Case Studies of Dealing with Study Design Issues. Before we get started, let me introduce myself. My name is Ronan Fitzpatrick, Head of Statistics here at StatSouls, who develop nQuery. I've been the nQuery lead researcher since nQuery Interim 3.0, many years ago at this point. And I've given workshops and talks at places like the FDA, JSM, and similar. And obviously give a lot of these webinars and demos as well for nQuery. So in terms of what are those extensions, those areas, those case studies we're going to cover today, well, I've split them into three parts today. Firstly, we'll be looking at the issue of non-proportional hazards in the context of survival analysis. Then we'll be looking at cluster randomization and newer extensions involving stepped wedge designs. And then we'll be looking at three-arm trials in the context of non-inferiority testing, and then a small time for conclusions and the discussions and replies to your questions at the end. Obviously, this webinar is being presented with nQuery, your number one solution for optimizing clinical trials. And this complete trial design platform will help make your clinical trials faster, less costly, and more successful. And if, in 2019, 90% of organizations with clinical trials approved by the FDA used nQuery, and you can see some of the reviews here, and also some of the companies that use nQuery along the bottom there as well. So with that out of the way, let's get into the meat of the webinar. And as I said, these are case studies. So what I'm hoping to do is kind of show, you have your standard method, let's say your log rank test, let's say your t-test, and what are some of the complications or design issues that could cause you to reconsider whether that design or test is appropriate, and what kind of adjustments are then required when you're talking about, say, stuff like your sample size calculation, which obviously is of high interest because nQuery is one of the leading sample size determination software in clinical trials. So this first area we're going to look at is non-proportional hazards, which is relevant for survival analysis. Now, I've covered survival analysis in depth in several webinars in the past. So if you're interested in knowing more about sample size for survival analysis, we'd be happy to share the recording of those on request. So feel free to get in touch about that. But in short, survival analysis is about, statistical inference about the expected duration of time to an event. And obviously survival analysis implies that event is often something quite serious, such as mortality or death, but also could be something like progression to the next stage of cancer, for example. And for this type of analysis, where we're trying to estimate the expected time until you have an event, there are many, many common methods proposed, but two most commonly used are probably the log rank test and the Cox proportional hazards regression model. Technically, the log rank test is really just a subset of the Cox model, but we don't need to go into that today. Now, the important thing to note about survival analysis in the context of sample size determination when we're designing a study is that the power is related to the number of events, not the sample size. Why is this? Because individuals who don't have the event will end up being right censored. And while I wouldn't say that they're ignored completely, of course they are used to adjust the analysis, they're not the ones driving the statistical hypothesis of how much time it takes to get to an event, because if you haven't had the event by the end of the study, then you aren't contributing a time that can be used to differentiate your control and treatment groups. And thus the sample size calculations that we'll be using both here for your standard methods like the log rank test, but for methods that apply for non-proportional hazards, the sample size is really just a determination of how many people do I think I will need to get that number of events. It's almost more like a meta model on top of your events calculation, which is the core of what you need to do. So if I require 100 events and I say I need 200 sample size, if it ends up that the recruitment is slower than expected, or the events are coming in slower than expected, then I can take steps to accelerate that to get more of the events that I actually need. And because of that, flexibility in the analysis and design of survival analysis studies tends to be much higher. And that will depend on many of the options about what we think is going to happen, and about how we've chosen to design our study in terms of stuff like accrual time, follow-up time, dropout, etc. So as I've mentioned, I've covered many of these issues in the past, in previous webinars, specifically on survival analysis. But the one that we're interested in today is a very common assumption for survival analysis and for the Cox regression model in particular, that of proportional hazards, which is basically that you have a constant hazard ratio between your two groups. So basically, no time variation in your treatment effect. Or indeed in any of your other effects that you choose to include as covariates. So lots of these other things like accrual patterns and follow-up and censoring and crossing over. I'm not really going to cover these in detail today because they're not the core part of this webinar, but as I say, if you're interested in these, I have covered these in the past, so do get in touch if you want to see our core material on survival analysis. So we're going to focus in here today just on the issue of non-proportional hazards. So as mentioned, the Cox proportional hazard model and implicitly the log rank test rely on the proportional hazards assumption, and ignoring this can lead to wrong inference because there's very different ways that this could occur. So when we have this non-constant effect size, this non-constant hazard ratio, then different things could be happening. So you can see here on the right hand side we could have a delayed treatment effect. This is very common in the case of immunotherapies, for example, where what they're seeing with these immunotherapies is that it takes some amount of time, a few weeks, a few months, where you see no difference between your immunotherapy and the proposed, control or reference treatment. But then after that period of time, you do start to see that significant improvement. And, that's obviously very different from the assumption that these two lines will start separating at a constant rate over time under the proportional hazards assumption. And you could show very easily that the Cox model or the long rank test will have, reduced power if you have this delayed effect. But there is also other major issues such as crossing hazards, where, the treatment effect basically reverses, where your treatment is initially, say, better in the early stages, but by the end is actually worse than the control group. And then, you could have a diminishing treatment effect or an increasing treatment effect, where it's basically more important or more valuable as you go on towards the end of the trial. Delayed effect is kind of a subset of the increasing treatment effect problem. So what you basically have here is a number of reasons why this is happening. As I say, immunotherapies, the leading hypothesis for why you get that delayed effect is simply that it just takes time to activate. There are other hypotheses, such as that this could be explained by immunotherapies being more targeted, that there's basically a subgroup where certain genotypes are better served by certain immunotherapies, because obviously a lot of them are developed in the context of being targeted for certain cancers or for certain people with certain genotypes. So that could have an effect where you could see a similar delayed treatment effect type hazard curve, but which is actually due to some people responding and some people not responding. And then sometimes there could just be other covariates or stratification effects that are causing this over time. And then sometimes just things are complicated. Sometimes you just don't get the kind of the constant effect hazard ratio over time. That just sometimes happens. And I think as we've seen a massive improvement in the quality of data for survival analysis over the last 20 years, this issue has been noticed a lot more as we've gone over in time. As you get more times that you follow up with patients, as you get more accurate results, they found that this NPH problem has become more significant. And because of that, we're seeing a very active debate about, well, how are we going to deal with this, both in the context of, preclinical research and just out there in the wild, but also in the context of clinical trials in particular. And so there's been a lot of different methods being proposed for this. And this is a very active area of research and of debate within clinical trials and phase 3 clinical trials as well. So none of this is certain. I'm not going to say any of this is like in stone, certainly not, and far from it. But there's lots of proposed solutions at the moment. So, there's like the usage of weighted linear rank tests, which is basically where events, depending when they occur during the study, are weighted higher or lower. So for example, using something like a Taron Ware or Guilhem Breslow, those are ones that tend to emphasize earlier events more than the log rank test or Cox regression model. But as I say, they're basically equivalent if you only have a single effect for treatment in a Cox model. That's a log rank test. And then there's been the suggestion of, well, take the weighted linear rank test one step further, where you actually combine the results from multiple linear rank tests and weight those appropriately based on what actually happened, and then use that to output a kind of, a robust result or statistic or inference about that. A lot of debate about that. Max Combo has a lot of interesting papers on it right now. And then the one we'll be focusing on today, which is the piecewise weighted rank test, which is basically an extension of the rank test, for example, the log rank test, but where instead of having a single weighting scheme, we also split our trial into distinct time periods where we actually explicitly say we expect different hazard ratios to occur and make inferences based on that. So basically weighting different like sub times or sub units of time of each study differently. We'll talk about that in a moment. But there are also, just for the sake of fairness, other model-free approaches that have many proponents, such as the median survival, just using that as your kind of measure of effect. There's Kaplan-Meier, KM curves, there's ways to directly compare those. And one of the ones which has seen a lot of traction in recent years is the use of the restricted mean survival time, or MST, as one of those ways of kind of comparing Kaplan-Meier, these non-parametric, the Kaplan-Meier is a non-parametric way to characterize your survival analysis data, and this is one way to compare two Kaplan-Meiers against each other effectively in a principled fashion. And this is an area where basically in nQuery we're going to be looking at this a lot over the next couple of releases, but we're just going to be focusing today on that piecewise weighted log rank test, which was included in our last update a couple of months ago. So what is the piecewise weighted log rank test? Well, it's basically a model that's been proposed to deal with the non-proportional hazards problem, and the version that we'll show today is specifically related to the delayed effect. So remember, the delayed effect is where for the first time period there's no difference between the control and treatment, but then after that there is something that looks like our constant, like proportional hazards type thing occurring, where this increases out here. So for this first period here, nothing's happening, and then suddenly you get something that looks kind of like your standard survival curve. And so, the piecewise bit is just saying that there's a different assumed hazard ratio per time period, namely in the simplest case where it equals 1 until some specified time, and then some constant hazard ratio from that point on. And then the weighted bit just refers that the different weights are applied to how important we consider the hazard ratio from each of those periods. And so the simple model that we're talking about here, what's referred to by Zhu et al. In 2017 as the Apple model, is one where we assume a hazard ratio from time 0 to time t, and then a constant hazard ratio from time t onwards until the end of the study, where we do right censoring. And that the weighting that we'll use is that we're going to give no weighting to the pre-hazard rate, to the constant, the hazard ratio equals 1 period. So basically we're not too worried about the first 6 months. That's just how it is. But then what we're really interested in is that, after 6 months, we're saying, for example, our treatment's going to show its efficacy. That's what we're going to focus on here and give that a weight of 1 post that time. And so that's a very simple model. You could obviously do a lot different weightings there. You could also assume different hazard ratios, but under this very easy, simple to understand Apple model, this is the starting point. And you can obviously use simulation to evaluate what would happen if we change these weightings, change these hazard ratios, like the null hazard ratio before t, etc. But it is important to note that no matter what you're doing in regards to those weights, you are having to make a fairly strong assumption about when you expect the treatment to start being efficacious. So you're having to say, I think after 6 months, after 12 months, after 3 months, after a week, we think the immunotherapy will kick in, for lack of a better term. And you need to know that a priori, and you need to build that into your model. Obviously, in the context of clinical trials in particular, you can't just make this up as you go along. These models have to be pre-specified. That's a key part of regulatory practice in terms of designing your study. Now, there's always, of course, wiggle room, when things just diverge away from what you expect. But, ideally, you would know and have specified using the scientific evidence available when you think this time when the immunotherapy will show its efficacy over the pre-existing treatment to occur. And much like the Cox regression model, you do have to make a fairly strong assumption about what you think the baseline hazard is going to be, because under the baseline hazard in the Apple model, that baseline hazard is going to be what's going to be happening to both groups. So you're assuming that both groups are going to basically have more or less the same type of hazard profile, and you're having to make an assumption about that. And that's fairly strong as an assumption. And then you have this random time lag model from Zhu that extended this to allow for time to vary, like the time till the effect kicks in, to vary by subject. We're not going to talk about today, but just be aware there is extensions for that. There is obviously been response to the criticisms outlined here, or some of the restrictions outlined here. But for today, we'll just focus on the simple one here. And if you're interested in how the test statistic looks, it looks like this here, with this SWS, the middle one here, being this simplified Apple model, which is just a subset of the much wider piecewise weighted log rank test statistic. So this is that subset where HR equals 1 and constant hazard ratio onwards, weight 0 before time t, weight 1 post time t. And in theory, this could obviously be extended to 3, 4, 5 different piecewise parts as well. But for now, we'll focus on the simple 2 one. So to illustrate this, I'm actually going to use a fairly, I'm actually going to extend a pre-existing example from the survival webinars I've done previously, which was looking at everolimus for advanced pancreatic and neuroendocrine tumors from the New England Journal of Medicine. And what I'm going to do here is basically start off with what they actually planned for in the study, which is where this particular treatment kicked in immediately, had a constant hazard ratio, but then effectively imagine that maybe this is an immunotherapy instead, or maybe this is what actually happened for everolimus, or what actually ended up happening. Obviously that it didn't, but just hypothetically, that instead of this hazard ratio of 0.6666 occurring from the start, let's assume that for the first 6 months, there's no difference in the hazards in each group, and then only after that does this hazard ratio of 0.666 kick in. So this is for a standard log rank SS analysis. All I'm going to do is extend this under the assumption that for the first 6 months, there will be no difference in our groups, and then only subsequently would we find this hazard ratio of 0.66. So we're really taking a pre-existing example and extending that. But to be fair, what I'm first going to do when I'm replicating this example is just make sure that we have the original example done correctly. So, this is nQuery. If anyone's not familiar with nQuery, the sample size calculations happen in this top left panel. There are help cards here to give you, what the definition of each row is, but it's also given on the far left-hand side what each row is, and each column is an individual calculation, with those rows in yellows being those that you calculate for, in this case, power and sample size. So let's do our calculation in column 1, where they had a test significance level of 0.025, and that was at the one-sided level. They had an accrual period of 74 weeks, but you'll have noticed that the median survival is given in months. In survival analysis, it's very important that all time-dependent units are parameterized on the same time unit. So we're going to take that accrual and follow-up period in weeks and turn that into months simply by dividing by 4. So 74 divided by 4 is equal to 18.5, and the maximum length of follow-up is simply the accrual period plus the minimum follow-up period. In this case, the minimum follow-up period was 39, so 74 plus 39 is equal to 113. And we take 113 and divide that by 4, we'll get 28.25. If you want to check the math, feel free to do so, but I have done this example a number of times, so I'm fairly confident on the numbers at this point. So the other thing we need to do is enter these exponential parameters. This is a model where we're going to assume exponential survival curves. Very standard assumption when we're starting for a sample size calculation. And for median survival, we're going to have 9 in group 1, which is the treatment group, and 6 for group 2. And we get this hazard ratio of 0.66. So we're using here the side table, which appeared automatically when we selected the exponential parameter row 1. And this is basically just a small tool to convert between different types of survival parameters. And we now have our exponential parameters from our median survivals. And we click transfer, and those move into the main table. And then we enter our power of 92.6, and we get 176 per group, which is equal to the 352 given in the researcher statement. So 352 patients, you can see that here. And then 282 events. So remember, we need 282 events. And under these assumptions about how long we'll be recruiting people and minimum follow-up, we're saying we would need 352 patients to get that number of events before dropout. If we want to count for dropout, it goes up to 392. Okay. So moving on, that's a fairly simple example. So now let's move on to the piecewise weighted log rank test. So as I said, like the major thing to take away from this here is that, now we're going to make an assumption that for the first 6 months, this difference did not exist. And in actuality, the exponential parameters were the same in both groups. Like it was equivalent to have a median survival of 6 months in each group, basically, for the first 6 months. But everything else will keep the same that's constant. So we'll keep 0.025 at the one-sided level, 18.5 accrual period, 28.25 maximum length of follow-up, power of 92.6. We'll have equal sample size per group. That was assumed from the last table. And so now we're saying, okay, what is the time to treatment effect? T1. That's our delayed time or delayed effect, whatever you want to call it. And in this case, we want to say that is 6 months. So we're saying that for the first 6 months, let's assume that instead of having an immediately hazard ratio of 0.666, that we had a reduction of 1/3 in the events in the treatment group compared to the control group. Let's say for the first 6 months, they're both going to be at the same hazard as the control group, because that's our baseline hazard. We'd assume is going to be roughly what will be in our control group. That's the assumption we're making here. And then we need to calculate the proportion surviving beyond time to effect P, which is basically just how many people will still be alive, just for simplification's sake, assuming everyone had started from the very beginning. How many people would be alive or not had the event by the time we got to 6 months. Now, thankfully, we have a situation here that if we assume that the control rate would have been, a median survival of 6 months, well, then if the median survival is 6 months in each group up until 6 months, then we would expect 50% of people to not have had the event. I think that should be relatively, the median survival definition is when will 50% of the events have occurred? If both are characterized by a median survival of 6 months up until 6 months, then we'd expect 50% alive. That's ignoring the accrual effect, by the way. Obviously, that would make things a lot more difficult to analyze, but that's ignoring that. That will obviously be accounted for in the calculation, but this calculation of this here, within the study, within this calculation here, within this little formula here, doesn't account for that. And then we have our post-delay hazard ratio, which we're going to keep as the same as before, of 0.66666, or 2/3. And you'll see here that we'll need 848 people per group. A very substantial increase, you can see, over the previous normal log rank test, which makes a lot of sense. Like if we're missing our effect size for the first 6 months, that's a very substantial amount of time where we're not seeing an effect. And therefore, obviously we're only really dealing with the last, in this case, around 18 months where we have the effect occurring. And so this sample size increase is very bigger than we would expect. But as we'll see in a moment, if we had this effect, if this existed and we had just used our standard log rank test, we would have seen a very dramatic reduction in our power compared to the 92% power we see here. And so that's, something to keep in mind, basically, when looking at this. So you can say we need 424 per group. So we're talking about, about a 200 and something increase over the previous example, not per group. So that's a non-trivial amount. Like we're nearly double the sample size, but that's not surprising for this type of effect. This is a very strong, very long amount of time to have this. And this obviously illustrates that immunotherapy is a more challenging type of treatment to evaluate fully, and maybe will require larger clinical trials. So that's maybe somewhat of a drawback for it. And indeed, the idea that it takes 6 months for it to kick in, if you're at a very severe stage of the disease, 6 months can be a very long time in practical terms. So as I said, though, if we assume the same thing, the same study design, but had continued to use the log rank test, what would have happened? Well, we can actually evaluate that in nQuery using simulation. So how do we do this? Well, basically what we're going to do is set up a design where there was no difference in the groups for the first 6 months, then set it so that the effect size, the hazard rate in each group would revert to what we had in our original design, and then see what would have happened. So I'll just set up our simulation design here. We'll just use a random seed here. And so in this table, we're going to set up a full set of what we think is going to happen in the study and replicate what is basically being assumed here. 6 months of no difference, followed by a hazard ratio of 0.666, which is basically equivalent to saying, exponential parameters of these two exponential parameters. So for the first 6 months, we'll set that end of time period 6 for column 2 here in the side table, 18.5 for here. This will just ensure that we have the same accrual period as previously, and then 28.25 for the end of the study. And then we're saying for the accrual, this is like we're assuming constant accrual up until this point, so let's keep that way. So 6 divided by 18.5 gives you around 0.324, so we'll give this as 32.4%, and then 67.6% for the second time, and then 0 accrual for this. So we're saying basically this is just the same equivalent to saying there was constant accrual from time 0 to 18.5, at which point we stopped accruing new subjects. And then we get into the dropout problem. Let's just set that to 0. We're not assuming any dropout in this particular example. So then let's set up what we think is going to happen. So we're saying that for the first 6 months, there's no difference, and that both of the hazard rates are equal to 0.116. So remember, that's equivalent to a median survival of 6 months. So we're just going to copy and paste this just to speed things up. And so in this case, like we're assuming that group 2 is the control group. So that's obviously going to be the same for the entire study in the group 2 row here. But in the group 1 row, the treatment group, for the first 6 months, it's the same. But for the subsequent 2 months, it reverts to what we expected in the original design. This hazard event rate of 0.007 of a median survival of 9 if we started from the beginning. Let's just enter that here. And then let's put in our sample size of 424 per group and see what power we would have got. So it'll take just a few moments to run. And obviously we could replicate the original log rank test, but that's relatively trivial here. But you can see that if we had used the standard log rank test under the same assumptions that we had used to define the sample size using the Apple method, using this fairly simple Apple model, we would have only had power of 57%. Only a 57% chance that we would have a significant p-value given this existed if we'd use a standard log rank test instead of the piecewise weighted log rank test. So obviously this Apple model is fairly simple and fairly restrictive, but if this is what actually happened, you're seeing a very, very, very large increase in power. And we could, let's just decrease this to 1,000 just to make this simpler. Like we could see like roughly how much sample size would we have needed to get to an equivalent power as the Apple method under this assumption by plotting it out here. So this will take a few moments to run. So remember, if this is a standard log rank test, obviously you're better off using, a log rank test if you have a constant hazard ratio. That's the assumption of where it's the most powerful test. The Cox regression model is basically the same thing. But if we had this delayed effect, this model suddenly becomes a lot stronger because we'd only have around 50% power for the same type of results here. And that's, I can just leave this here for a second. And we probably need to increase the sample size significantly to get back up to that result. Okay, I think that's enough here on that. So hopefully you've learned something there. And as I say, there's a lot of other models proposed for the same problem, like the max combo, these weighted linear rank test, median survival, or MST. We'll be looking at all those in future releases of nQuery. If there's particular solutions that aren't mentioned here or ones that you think are particularly interesting, do feel free to get in touch, and that will help us prioritize which ones to look at sooner rather than later. Okay, the next section we're going to move on to is cluster randomization. So cluster randomization is where, the treatment is being randomized by cluster, not subject. So, let's say we have 100 people, and there's 10 schools. So, we have 10 people per school, 10 students per school, or probably per class in this case. And we have two options to randomize. We can either take the full 100 people, and then regardless of which school they're in, we'll put 50 into the control group and 50 into the treatment group. This is fully randomized design, probably the default in a clinical trial. But maybe that's difficult. Like if you were doing a teaching intervention, it's kind of hard to take like 3 students in one class and give them one teaching method, and then the other 7 a different teaching method. They're in the same classroom, so you're we'll say impossible nearly to do that. So what you have to do is to randomize, 5 of the classes of 10 students to the treatment group, and then 5 of the other classes to the control group. And that's where you have cluster randomization. I think the important thing to note is that if you do that, the effective unit of inference really becomes the cluster. This is kind of emphasized by the Nelder or Rothamsted school of approach to inference, which Stephen Sen talks about a lot here, and this is where his screenshot is from, for cluster randomization. So, it's important to emphasize that if you move up one level in randomization, that directly affects the causal, framework for how you should interpret that study. Like the causal level is really at the cluster level at that point. Now you can make subject level inferences using some assumptions, but the cluster is really the level of causal inference in this randomization scheme. Like basically causality follows randomization is more or less the idea of experimental design. But at a practical level, if you're going to use cluster randomized design, you're not getting something that's really fully randomized. You're getting something that's close to, hopefully, but not quite that, because obviously, imagine that, 5 of those are boys' schools, 5 of those are girls' schools, but like 3 of the boys' school, like 4 of the boys' schools end up in one side and only 1 on the other side. That increases, probably many of the imbalances in that. So what you need to do is adjust for the fact that within clusters, you're going to see more self-similarity than between clusters. So, a boys' school in a poor neighborhood, the students are probably going to do more self-similarly than, a girls' school in a more socioeconomically privileged group. And therefore, you need to adjust for how self-similar those centers are. And you can also do a direct adjustment for those covariates as well, of course. But, you just need to ideally account for the intrinsic self-similarity effect using a measure such as intra-cluster correlation, the coefficient of variation, or the within cluster variance. And you can actually show most of these are relatively interchangeable with each other. They're basically interpreting the same idea, but from different directions. And as said, these are useful mostly if it's difficult to randomize by subject, but also can sometimes lead to reduced costs. And if there's, as I say, a contamination effect where, it's difficult to splice out, 5 people within a cluster to only get one treatment and 5 get the other, because they share, say, the same space, like, they share the same hospital rooms, the same school rooms, then that contamination effect can be, undermine a fully randomized design, and therefore you may need to go to cluster randomization. But you do this at the cost of, really lowering your overall power / effective sample size. And then there's obviously that problem of selection bias and basically a higher chance of ending up with unbalanced designs or randomization because you're moving multiple people at the same time rather than each person individually. And then so that's like what I've mostly talked about there is the simple cluster randomized design, but there are extensions of this, such as the stepped wedge design, which is where subjects move from treatment to control over time. So let's say like you look at this pattern here. What we're saying here is that in time point 1, say after the first month, everyone's getting the control group. In time 2, suddenly group 6 is getting the treatment group, and then over time that's moving up and up and up until everyone is getting the treatment group, almost like a kind of crossover type design where everyone has to control at baseline. But this is a design that's often paired with cluster randomization. Once again, the unit is the cluster, not the subject. And this is one reason why you might want to do this is because of treatment scarcity. So imagine you want to teach a new new nursing technique. You probably not have that many people who are capable of teaching that method, right? Because it's a new intervention, like you're trying to evaluate this new intervention. And therefore, you only have one team to teach. Like they can't be in, 5 places at once. So they need to teach in hospital 1 for the first time, like for the first month. And then only in the second month can they move on to time 2, cluster 2, hospital 3 in month 3, so on and so forth. And that's naturally creating a stepped wedge design. And that needs to be accounted for in your modeling, which you can kind of see on the right-hand side, and in your statistical analysis and power analysis, etc. And there is one nice thing from a statistical point of view, though, is that you are getting this within cluster analysis. Similar to a crossover trial, you do now have an estimate of, like, you can compare, a cluster, how it was doing before the intervention and after the intervention directly. So you're getting the within cluster analysis as well as the between group analysis, which is some things. But obviously there are some issues here where the allocation bias, the expensiveness, being expensive, and the complex analysis all may mean that this is not viable for your particular study. So, cluster randomization and stepped wedge cluster randomization, these are really tools usually used in response to real practical problems or realities that you need to account for in your statistical analysis, because that's just how it has to run. And so what we're going to do here is actually going to do a stepped wedge design, which was looking at the membrane sweeping to reduce induction of labor, basically teaching midwifery teams this, the NICE guidance. And this is a relatively simple cluster randomization where they had these 12 weeks, where each week they were moving on to a new set of midwife teams. They had 10 hospitals that they were looking at, and then 12 measurements per cluster per time. These measurements were basically 12 births per cluster per time. There is like one small complication here that we're talking about, what's known as a cross-sectional design, where there's 12 new subjects being looked at each time. So we're doing the same cluster group multiple times. In this case, we're looking at new measurements per time. Thankfully, in the case of births, we're probably not expecting old births from the last week to be new births this week. That's something strange is going on if that's occurred. So thankfully, this is naturally a cross-sectional design where the stepped wedge is probably best understood. And you can see that within clusters, we're not expecting a huge amount of self-similarity. So we're saying that more or less hospitals are mostly interchangeable with each other here. And so they wanted to know that they'd have a baseline proportion of 0.4 in terms of the endpoint of interest, and they wanted to increase that to 0.5, basically. Now, technically, they looked over a wide range of different scenarios, but we're just going to look at this specific scenario today. And so we can just go to the stepped wedge study here. So what we're going to do is, it's firstly actually replicate something close to what they had there. So they had a 5% or 0.05 significance level at the two-sided level, a group 1 proportion of, say, 0.4. We're assuming group 1 here is the control proportion. And in this case, they actually calculated what would be the greatest effect size they could see given the constraints that they had in terms of access to nurses and births and so on and so forth. Apologies if there's any background interference. I live near to a major traffic thoroughway. So they did an ICC of 0.02202, and I'm not going to go into this much today, but basically complete means that you have that perfect one for every time unit. You basically step up once. Whereas if you don't have a very specific relation between the number of time periods and the number of clusters, basically k divided by t minus 1 equals an integer, you can't get a perfect step up. At some point you have to skip one and do the same, not move on to the next group for 2 weeks or 2 time periods instead. This isn't a balanced design or a complete design. It's actually a balanced design, which means that in this case we need to just set the balance here, because if it's complete, then you have to fulfill this k minus t minus 1, they divided by t minus 1 equals an integer criteria. And so they had a number of time periods equal to 12. They had 12 measurements, 10 clusters, and 12 births per time period, basically, per cluster per time period. So in each cluster at each time period, 12 births would be evaluated. And then they had a power of 80%. Now, I'm just going to manually select the correct solver here to ensure that, we're going to calculate pi2. But we want to ensure that this gives us the correct one here. That happens to be this. We're going to calculate given pi1, but we want pi2 to be greater. So we enter 80 here, and you'll see here that we get around the correct proportion here. So they said it would be about a 10% increase, or 0.1 increase on the proportion scale. You can see it's actually closer to 0.114. Take 0.514 minus 0.4, that's around 0.114. But the thing is that we didn't fully replicate what actually happened in the study. Because what actually happened in the study is that they had a pattern of results that looked like this, where in actuality there was a lead-in period where basically they didn't do anything, they didn't change anything, and only after 12 weeks of lead-in did they do the intervention, and then follow up for 12 weeks where the intervention happened. And for practical purposes, we can basically ignore the lead-in period. So imagine that the study really started here at week 13, and then went on to week 39 here, where you can see here there's not even a very even step pattern. So you can hear, before they got to group 3, there was a week where they didn't do any new intervention. There's a gap of week 2 here. So if we want to really replicate what happened, we'd have to enter a recruitment pattern that replicates this, and that is possible to do in nQuery. To do that, all we need to do is to replicate what we put in previously, but instead of selecting a balanced design pattern, we need to enter a custom design pattern, which is the last option here in this drop down for design pattern. But to also account for the fact that there's going to be time periods where nothing was happening, because these white time periods are equivalent to missing data effectively, we're going to basically assume that the length of time was from 13 to week 39. And that basically means that we're going to assume that there is 27 time periods, including the 0 time period, of course. We're including one time period where everyone didn't get the intervention. So it's really starting from week 12, technically, this week where nothing was happening for anyone, theoretically. And then we still have 10 clusters, and we still have 12 per time period. But basically what we need to do is basically just, for any time period that's in white in that previous pattern, we're just going to consider that to be equivalent to missing data. And so to do missing data in nQuery, we just enter -9 into the cell manually. You could use, equal 9, into the time here for no observation. Now, obviously, I could write this out manually, but that's probably a bit of a tedious thing to watch. So I prepared this earlier. So you can see here that 0 indicates that you were in the control group, 1 indicates that you were in the treatment group, 9 indicates that there was missing data here. So the group that, or cluster that moved into the treatment group first has these missing data where basically the study had stopped for that cohort. Whereas for this last one, you can see that it went, once it had finished all of its observations, the 12 weeks of observation, that's when it was finished. And that's when the study ended. So we're just going to put that into nQuery in the side table here. Oh, and we have the wrong solver here again. So let's just select solver 3 here to get the correct result where the treatment group is better, has a higher proportion of success. And you can see there in that case, now it actually equals exactly the 0.1 effect size that the authors had. And we could compare this to what would have happened if we'd used a standard, study design where we had no cluster randomization. So let's say, 0.4 and then power of 80%. And, this sample size, what was the sample size in this case? Well, it's 12 by 10 by 12, 1440. If we take 1440 and divide that by 2, we should get something like 720, I believe. And, in that case, if we had just assumed that this wasn't a cluster randomized problem, we would have been over optimistic. Like we would have said, we could get effect sizes as low as 0.473. And that would have obviously been a major mistake on our part. And if we had looked at, say, a normal cluster randomized case where we ignored that effect, we could have ended up with a very different study. So let's imagine we don't have repeated measures. Let's just say you only did analysis in each cluster once. Let's assume we had the same effect size. What would have the sample size required have been then? Let's assume the simpler case where there's just 12 looks per group, and then look at a power of 80%. We would have need 36 clusters in that case. A lot more nurses would have, a lot more hospitals would have been looked at. But maybe, the more equivalent sample size is the 144. 12 by 12, 12 sample size per time period by the 12 by the 12 time periods. So we've looked at each nursing school 12 times and done for each time period done 12 in individual ones. And you can see that even if we did 144 looks per each cluster, we would have required 11. We would have required slightly less here, but once again we would have been over optimistic effectively, which again would be highly problematic. This should be 0.2, apologies. So that's my mistake. So you can see here that, you'd need 40 per group, and that's a lot more clusters, and then this would have been slightly less efficient than using the, the step wedge design, that if we just consider these as 144 individual measurements. So, step wedge design, it's certainly not a free lunch, but where there are important things that it deals with, it is important to use an analysis that reflects the reality of your study. So there are multiple complications associated with those, which I covered in a previous webinar. If you're interested in these covering more, feel free to get in contact. But basically there are additional complications that come with it. But if you're having this effect where it's taking time to get to each cluster, a stepped wedge design might be you. And if cluster randomization is a constraint due to contamination or other effects, then you should definitely adjust for that cluster randomization. If you ignore it, you're going to be way too optimistic about what your study is doing, and you'll make mistakes about your sample size calculation and end up being underpowered. So the final thing I want to cover here, relatively briefly, is just three-arm trials, because this is mostly just an extension of your standard non-inferiority testing, where non-inferiority test is where you just want to prove that your treatment is no worse than standard by a pre-specified margin, known as the non-inferiority margin. And this non-inferiority margin is often selected as a fraction of what's considered to be the active control effect. So basically, how well did the standard treatment do versus placebo? You want to take a certain percentage of that M2 as your non-inferiority margin. This is talked about a lot in the FDA guidance on the non-inferiority testing, which is included in the reference here. And obviously, non-inferiority testing is very common in the case of generic drugs and medical devices, where usually, you're not making a claim that your drug is better than the pre-existing drug. It's a generic, it's just, the same thing as the pre-existing drug, you're hoping. or a medical device, a pacemaker. You're not expecting a pacemaker to revolutionize that problem. You just hope it has comparable performance to other things. And so you need to control, you're just comparing treatment to control without placebo. That's a standard non-inferiority test. But the problem with that simple design is that you haven't really proven within your own individual trial that, the reference treatment or indeed your own treatment are significantly better than placebo. That's been assumed based on the prior evidence, but it has not been proved by your own study. And in effect, you're basically using something that's equivalent to a historic control. And that means that when you're doing a non-inferiority study, what often is required is a very stringent replication of what occurred in the original superiority trial to ensure that the conditions are exactly the same, and so that you have comparable effect sizes occurring for both the reference treatment, the ORLD, and the the actual new treatment, the generic, for example. So if that's a major issue, and it is something that is a major issue if you go into the guidance on this, what is one solution? Well, one solution would be to, rather than having to replicate a prior study, why not just include a placebo arm directly? So bring back in the placebo arm and then compare the placebo arm to either and or basically the reference group or the experiment and or the experiment. And then if you're doing better than the placebo group in your 3-arm trial, then obviously now you have shown or proven that not only is our treatment better than placebo, but then when we do the non-inferiority test, then we've also shown that the treatment is equivalent to reference. And this is why it's often considered to be the gold standard. The reason it's not done more often is the simple reason that placebo would not be allowable if it's unethical. Like if we have a reference treatment that already works, and this is a serious condition, for example, we can't give people placebo because then we're exposing people to a high risk. But in certain areas where treatment effects are more uncertain, or there's more uncertainty in general, or the reference treatment is fairly ineffective at this point, then placebo then becomes more acceptable in those cases. So if you can do this ethically, there's no reason why you can't do that. And theoretically, what you just need to do if you have a placebo group is, one, test the hypothesis that your experiment or your reference is better than placebo, and then test whether the experiment is non-inferior to the reference group, basically an hypothesis test greater than the non-inferiority margin. And because that's a what we call a conjunctive hypothesis, like both have to be true to prove your analysis. Like you can't, if I prove the first but not the second, then I can't do it. Basically a gatekeeping procedure where hypothesis 1 has to happen before I'm even willing to test hypothesis 2. In this case, H1A before I do H1B. Then we retain alpha without any multiplicity adjustment and stuff like that. The alpha is retained because of the very strict gatekeeping procedure. But one thing that's been shown by some of the theoretical work in this area is that you can simplify this down to really a test of the ratio of differences. So the difference between experimental and against placebo, or E minus P, divided by the reference versus placebo. So, the effect size for the experiment over placebo divided by the effect size of reference over placebo, and then showing that is greater than some non-inferiority ratio, which we call theta here. And so this is what we call a complete testing procedure, which basically takes these two sequential hypotheses, can be basically talked about as if they were this one complete testing hypothesis here. And that can be evaluated in a unified framework using a wall type test for a variety of different endpoints, from means to proportions to survivals to incidence rates and so on. And doing that, and then there's also ways that we can optimize our allocation to maximize our power given this 3-arm trial design. And as I mentioned, this has been extended for a variety of different endpoints by different authors over the last 20 years or so. And so what I'm going to do very briefly here is do a sample size calculation that has this 3-arm rule where, they actually had difference from placebo. So we're going to assume the placebo arm is 0. And then we had a 1.56% increase for both groups. So they actually are going to be equal under the alternative hypothesis that these authors assumed. And they had a non-inferiority ratio of 0.5. So they said that, if the experimental arm did no worse in terms of being better over placebo of 50% or 0.5 of the reference arm of the standard treatment, they will be happy enough to say it's non-inferior. And that's a relatively liberal one. That's not one you would usually see in the case of like drugs, for example, where this would be more stringent NI. But we're talking about here more of a particular a solution which is a lot less invasive than the previous one, the previous approach to doing this. So obviously the improvement in safety or the improvement in people willing to do it, that can sometimes justify a more or less stringent non-inferiority margin. Standard deviation of 2.5, power of 80%, and they allocated 0.38, 0.38, 0.24 in terms of experimental reference and proportions. This is the proportion in each of those groups. So with nQuery, this is actually relatively easy. So we just have our significance level of 0.025. And note that they say two-sided confidence interval of 0.05. Non-inferiority hypotheses are really at the one-sided level, so I have that here, just if you're interested in that. We said 1.56% for experiment and reference, and a placebo arm of 0. Like, these are technically really the differences from placebo, but for practical purposes, for the means hypothesis, that doesn't make a difference. The non-inferiority margin is 0.5, so we're saying if the experiment arm was had at least 50% of the effect over placebo, of the reference effect over placebo, we'd be happy to say it's non-inferior. And then a common variance of 6.25. So standard deviation is what we gave, or they gave in their statement there, but this is the variance, which is obviously just the standard deviation squared. So 2.5 squared is equal to 6.25. We had our allocation ratios of 0.38, 0.38, and 0.24, and a power of 80%. And you see we get a sample size of 352. This is technically, these are 134 instead of 133, but that's probably just due to very small rounding issues versus this. I've verified this against other software, and this is the correct example for these allocation ratios. Perhaps they were using 0.37 or something weird and got their result, but this is the closest replication here. And basically rounding would explain most of what they saw in difference. So it's 352 versus 350 that they got, but I'm pretty confident that's basically just a very small deviation based on maybe their allocation ratios having been slightly different. But, as I mentioned, there is an optimal allocation, which is really just saying an allocation that maximizes your power for a particular design. And in the case of the means case with common variance, it's actually very simple. It's given in the help card here. 0.5 for the experimental arm, the non-inferiority ratio divided by 2 for the reference arm, which is 0.5 divided by 2 of 0.25. And then that happens to be 0.25 for the other one here as well. And then if we enter 80% power, you'll see that the sample size requirement is 326 per group instead. Sorry, this seems to have gone a bit wibbly over a second here. Let's just restore this back to what it was previously. And you can see here that obviously the sample size is lower than the previous example of 352. So, if we wanted to simply reduce sample size, this would perhaps be considered the more appropriate allocation. And, if we were to compare that to a standard non-inferiority test, obviously now if we were using a standard 2-arm superiority test, we'd usually use the differences rather than the ratio of differences. So it's not necessarily as easy to kind of see exactly what that is, but basically we could see that, for this case, a difference of half 1.56 is 0.78. And therefore the expected difference is 0. We're obviously expecting it to be the same under the alternative. So this is just to note that this is, 0.156 divided by 2 because the non-inferiority ratio is 0.5. So that's a relatively simple calculation in this case. So that's what the difference would be under the null. And then a standard deviation of 2.5. We're back in the standard deviation scale here, and a power of 80%. And you'd have 163 per group here, which means would there be less sample size in total for this design, of course, but you can see that the sample size per group would be less for equal sample size allocation for E versus the experimental and reference group, which is the most common way it's done, even though this is technically the optimal design. So 2, 2, 2, 2, 1 or ones like that are the most common for this type of design. And so you can see here 163 per group versus 134 per group. So even though in total, this is obviously adding the placebo group means that you're looking at more people in total, you need less in your experimental groups. The placebo group maybe in certain cases could be seen as being less difficult to kind of deal with. Okay, and I think that's pretty much our time up. So I'll just get into the conclusions now. So trials will often require adjustments from our standard methods, whether we're talking about your standard t-test, your standard log rank test, your standard non-inferiority design, our standard fully randomized design. And if you have complications, you need to adjust for them. If you have complexities, they have to be adjusted for in your design and in your analysis. If you don't do so, it's very likely you'll have an underpowered study, you'll have a higher chance of mistakes, and you'll end up making, like having drugs thrown away that shouldn't be, or coming to erroneous conclusions that it's effective, and that's obviously not acceptable in the case of any type of study, but especially in clinical trials. So in terms of non-proportional hazards, this is a very common issue, especially in immunotherapies, and we should look at, and there's a lot of discussion now about weighted models, max combo, piecewise weighted linear models, and model-free measures such as the restricted mean survival time. We looked at the weighted models approach today. Cluster randomization is a very real constraint in the real world. For example, vaccine studies often deal with cluster randomization because it's easier to vaccinate a whole school than to vaccinate 50% in a school and then not vaccinate the other 50%, or give them a fake vaccination, or not fake, but a different placebo vaccination. And you need to adjust for the complex cluster effect, or for the cluster effect. And if you have a more complex design, like a stepped wedge design, you need to adjust for that too. And a 3-armed non-inferiority trial gives you a direct comparison against placebo for both your treatment and efficacy, dealing with the assay sensitivity problem, and be aware that there's a nice, flexible, easy to understand framework to do testing in that case. This isn't a huge leap forward in terms of doing the testing. It's actually relatively simple. So don't be afraid of the statistical side if you choose to go with a 3-arm trial. So that completes our webinar today. I just want to thank you so much for listening. I hope you have learned something useful today. And if you have any questions after the webinar, feel free to email us at info@statsols.com. And just to let you know, a couple of months ago, we released our summer 2020 release, which included 26 new tables in these areas. And if you want to try anything you saw here today, whether that be something you don't have access to nQuery, or you want to access something like the adaptive features in nQuery Pro, feel free to go to statsols.com/trial, and you can try nQuery with all of its features activated within your browser for a couple of days. And if you want to get started with nQuery and find some of our videos and former webinars, you can go to statsols.com/start. And if you want some of the references, they are available in these slides, which, as I said, the recording and the slides will be sent to you after this webinar is complete. So I'm just going to take a moment here to look at any questions that have come in during the webinar. And as I said, any that I don't get to today, I'll make sure to get back to you via email in a couple of days. Okay, there's just a couple of questions here. I won't get to them all today, but one was just about extending the non-proportional hazard models to deal with interim analysis. That's probably something that's going to happen fairly soon, I would imagine. And technically, you can do that already for a log rank test. You could simulate that using a log rank test at the moment, but obviously, as we said there, the log rank test is inefficient for that current case at the moment. But there is work happening on that, I would assume, and as soon as some of that research is made available, I think I've actually seen a few papers on already. Once that kind of comes more valuable, it's obviously something that we'll definitely look at. I suppose the delayed effect does make the how do you infer or make inferences about the early interim analyses work. There's practical complications there, so I'm very interested to keep reading up on that area, but it's not something we're looking at probably in the immediate future. And then there's just one question here about stepped wedge design, like how, I need to understand this question, but basically just asking, why did I include all those basically missing data? As I said, they're not really missing data. What we're trying to replicate here is the fact that this study design was kind of done in a certain way, which is to say that they had a 12-week lead-in period before they did anything. Then they did the classroom intervention for these nurses, for these midwives, and then they followed up for 12 weeks afterwards. But like, if this starts at week 13 was the first intervention, but they only followed up for 12 weeks regardless. They didn't do up until the end of week 39, only for 12 weeks after the intervention. You can see there's these times here that, for the first cluster where they weren't being followed up anymore. So that data is effectively missing. This isn't a problematic missing data problem, really. And that's adjusted for in this stepwise design. So basically, it's kind of trying to replicate what really happened. And based on the result that we got, you can see that's probably how they were doing their sample size calculation. Okay, as I mentioned, there's probably a few other things here, but nothing too important. I'll get back to you by email. So I just want to say thank you so much once again for attending. And as I said, if you have any questions, feel free to email info@statsolve.com. So thank you so much for attending, and I hope to hear from you soon. So once again, thank you so much, and goodbye.